Hello, dear colleagues, dear guests and attendees of the Precision Medicine Networking Forum. A little bit later, 2 minutes later, but we are starting our session of the Functional Precision Medicine. I am very glad that for the third year in a row we have such a session, and the aim of this session is usually to show that there are more tests available that can help even more to choose the right treatment for the right patient. And today I have a really wonderful co-chair, also annually, like we already are getting used to be a co-chair and chair. It's Caroline Heckman. She's the scientific director of the Institute of Molecular Medicine in Finland. Yes, and my name is Inese Cakstina-Dzerve. I'm a lead researcher at the Riga University. And as a first speaker, I would like to introduce you to Alice Soragni. She's assistant professor from Department of Orthopaedic Surgery member, Molecular Biology Institute member, Cancer and Stem Cell Biology, University of California, USA. And she will be talking about the models and how we can use the patient-derived models to look at the drug vulnerabilities. So,
thank you. Well, thank you very much for the kind introduction, and thank you for being here. Just so you know, the timer has not started yet. So, you know, the type of precision medicine that we do in my laboratory is a little bit different than the genomics-focused precision medicine you've seen so far, but I will tell you a little bit about that as we move forward. The way in which we apply our concept of functional precision medicine is truly focused on rare cancers and rare tumors. And the reason why we try to, develop novel models for this type of cancer is that, there are a lot of issues when patients come to the clinic and they are diagnosed with a rare cancer. And although, rare tumors are individually, of course, very rare, right? They happen in a very small number of people. Once you put them all together, they account for up to a quarter of all cancer diagnoses in the United States. In the United States, that comes down to 400,000 people a year, which are diagnosed with a rare tumor. So it's a lot of people for which we need to do better. For a lot of rare cancers, although not every rare cancer is the same, some we know well. The majority, we have very little data. We don't know what drives these tumors. We don't know how do they respond to therapy. For some, there is actually no standard of care whatsoever. And there are, of course, incredible challenges to running clinical trials which are timely and adequately powered. So it can take you 10, 20 years, right, to accrue maybe 100 patients so you can prove a basic intervention. So obviously, lots of challenges which are truly compounded by the fact that for a lot of rare cancers, we have no model. For a lot, we don't even have established cell lines, let alone, some of these more advanced patient-based models. And that's why we are trying to investigate this type of tumors more. We want to develop better models to aid in the drug development process. We want to develop solutions for these patients. We want to learn more about the biology of these tumors. But most importantly, we want to develop these models in order for us to implement precision medicine solutions. And so what my laboratory does is we work a lot with cells coming directly from patients, right? When you take a tumor cell from a patient and you don't want to modify it to force it to grow as a cell line, you actually only have a couple of options to grow these. They typically don't just grow on plastic, right? So you can take these cells and put them in a mouse to generate a patient-derived xenograft. Now, there is some beautiful work done in these models. However, from our perspective, which is, trying to screen these, to test hundreds, potentially thousands of different drugs and so on and so forth, these models are limited. Firstly, you cannot generate this for every single tumor. You typically can only generate this for tumors that grow really quickly. It can take months, up to sometimes even a year to grow and establish in a mouse. And then again, just for the sake of money and time, you can test very few interventions. There is also, something to be said about the fact that human tumor grown in a mouse will change. They will evolve, they will adapt, you will have selection going on, and so they become more and more different from the patient tumor you started from. The absolute easiest thing you can do when you take a solid tumor from a patient to grow that ex vivo is to develop what we call tumor organoids. So you take your tumor cells, you embed them in extracellular matrix, which is basically a jello-like protein-based substance, which is akin to how they grow in the body. And you can do these very quickly, very, very rapidly, with a very high success rate. And this type of models have, in principle, the ability to be screened quite broadly. Over the past half a decade or so on, we have accumulated a large body of work showing that this type of patient avatars are really good at recapitulating patient drug responses. And so that is why we think these are good models to try to enact precision medicine solutions. For the sake of our discussion today, I wanted to mention that there are different ways to develop organoids. Not everyone that tells you they're working on organoids are actually working on the same thing. The most conventional way to perform patient-derived organoid work is this long-term patient-derived organoids. So you take a tumor cell and you develop patient-derived organoids, and then you keep growing it, and passaging it over time until you have a established, resource. Now, this is great because it affords you, it's a tool that you can share with the community. It's a tool that allows you to study the biology in big numbers. You can get big numbers of cells. However, just like when you're growing these cells in a mouse, you end up selecting, right? And you end up purifying just for a proportion of the tumor cells. Again, there is a lot of pros, but, for us, we want to be as close to the patient as we physically can. And so we actually use this type of short-term or unpassaged, or some people will call it passage zero organoids. You take the cells, you put them in a matrix, you develop these organoids, and you never passage them. You very rapidly perform your drug testing. When you do it in this way, you have very high success rates. This works virtually almost all the times. For some tumor type, we have 100% success rates, right? They always grow like these. And most importantly, because the timeline is so short, it's about a week time, you don't have a lot of evolution, you don't have a lot of selection, but in addition to the tumor cell population itself, you also retain every other cell that is in a tumor. Now, a solid tumor, is tumor cells for sure, but it's also a number of different components in the microenvironment and support cells that are critical not only to the tumor biology and to the tumor growth, but are also critical to drug responses. And so we want to retain the ability to have these cells within our organoid models. Now, the disadvantage here are obvious. This resource cannot be propagated, and so you are absolutely and critically dependent on the cells you get from surgery. As I told you, these models are screenable. That is in principle true. In reality, there are a lot of technical challenges when you're trying to perform drug screening in 3D. And mostly because, you want the ability to use robotics and automation to perform large-scale screenings. And what these machines are great at doing is they're really great at going to the center of your well and kind of adding media and drugs and so on and so forth. But in that process, they can pluck things out. And so, conventional 3D culture uses domes of your extracellular matrix and tumor cells at the center of your well. This is one well of a 96-well plate. For us to be able to implement automation, we decided to reverse the geometry. And so we grow our patient-derived tumor organoids, is a pancreatic cancer case in this example, at the periphery. And so we leave our center empty. We develop these mini rings, and this truly allows us to get a nice model that generates very reproducible and robust data. Our robots don't pluck anything out. There is nothing to be plucked at the center. We have a very thin, even layer of extracellular matrix component, which allows nutrients and drugs to penetrate optimally. And we don't need a lot of cells, so really we don't need to do any type of passaging. Our timeline is very quick. We get cells from surgery, from biopsies, from ascites, from any type of procedures. We dissociate the tumor, we generate our rings, and then we let cells establish as patient-derived organoids over the course of about 3 to 4 days. We then perform 2 drug screening over 2 consecutive days. So we add drugs, and then we remove and add drugs again the day after. And at the end, we perform a final assay. We actually also do imaging over the duration and then use machine learning-based algorithm to calculate how these organoids are growing and reacting to drug treatment. Now, the formation of this type of ring structure was originally done manually, but as I keep saying, for us to get very robust data we can actually base clinical decision on in the future, we need to make this as robust and automated as possible. And so the next step for us was to take away the operator even from the process of generating the rings. And so now we have bioprinters that can print our tumor cells directly in the structure we want, which again increases the robustness of the screening and the data we can generate. We do have a lot of different projects on a number of different rare cancers, but the one I wanted to highlight today, because it's the one where precision medicine in our, has pushed the furthest, is sarcoma. Now, sarcomas are a family of rare tumors arising from tissues which are non-epithelial tissue, from muscle, from connective tissue, from bone, from cartilage. They account for about 1% of all diagnoses, but in that 1% little slice, there is actually, over 100 different subtypes. So it's an incredible diverse group of tumors. They are all very, very rare. UCLA has a very large multidisciplinary sarcoma clinic where we see almost 1,000 patients every year. So we have a really, truly high volume team of clinic and a team of physicians across discipline that work closely with us to make this happen. The timeline is just the one I showed you earlier. So we receive samples, from any type of tumor through biopsies or resection largely. We digest the tissue, we generate our patient-derived organoid models, and then we perform all sorts of characterizations, which I'm going to show you in a second. Our very first study was published about a year ago, where we showed our experience with the first 126 consecutively enrolled sarcoma patients, from which we obtained close to 200 samples. That is both because we have patients we follow over the course of the disease, and so you can get a biopsy, a resection, and then metastasis, but also because the particularity of sarcoma is a disease that metastasizes to the lung, and typically patients get multiple nodules within their lungs, and we can get individual nodules and study this separately. For that study, we screened about 29,000 wells with an average of 117 drugs per sample. When you look at the distribution of the different diagnoses, we covered about 24 different sarcoma subtypes. And, a couple of the characteristics of this cohort is, again, sarcomas include bone tumors. Bone tumors, albeit rare, are actually fairly common in the pediatric and young adult population, which is why about half of our cohort is younger patients. There is a slight biological preponderance for males, which is also represented, not very well understood, but certainly represented in our cohort as well. And then because we tend to be a reference center, we see a lot of patients which are actually already diagnosed and have been, maybe treated elsewhere for their primary disease. And so majority of our cohort is metastatic or recurrent cases with samples and patients that have seen a number of different types of therapies, including a proportion that has received radiation, which is typically done particularly for soft tissue sarcoma. I will say that although that is where the study was about a year ago, we are now, we have a very large biobank with over 1,100 samples across tumor types. But as far as the soft and bone tumor tissue biobank, we have now over 700 samples from 420 unique patients covering over 100 tumor types from over 500 surgeries. So a lot of distribution and a lot of, different subtype within sarcoma subtypes. So we think that is a quite unique resource we have available. Now, what is a sarcoma organoid and how does that look like? Well, the point is that, as I told you, there are 100 different types of sarcomas. They are very diverse between each other. And you can see a few examples here. And what we want is the ability to recapitulate the features that each and every subtype has. And so, some sarcomas are small blue round cells, and this is what we want to see in our patient-derived models. Some are spindle cells, and so we see that. But also, one of the particular, features of sarcoma is that not every tumor is highly cellular. Some are, tumor cells scattered within a matrix. And so, what we want to do is we don't want to generate just a tight ball of cells, right? That's not what the tumor is. That's not what the biology of the tumor is about. So we want for these tumors to have the ability to recapitulate those features. And so if it's scattered cells within a matrix, we want to have the ability to recapitulate that, which we can do quite well. We can also recapitulate the molecular features that are cardinal to the disease. This is an example of a patient with a well-differentiated liposarcoma. These patients are oftentimes diagnosed clinically in the US for the presence of an MDM2 amplification. And so this is how this patient was actually diagnosed. And we decided to look if the organoids retain this feature. And so we actually performed FISH on the organoids, and we could see that this MDM2 amplification, you can see this in pink here, was maintained in the organoids as well. In more detail, we can look at these, for example, at the whole genome sequencing level. We perform deep whole genome sequencing at 80 to 100x and truly try to look at the clonal and subclonal distribution of these tumors. This is an example of one of our children with osteosarcoma, a biopsy and a metastasis from the same patient. And so you can see, interestingly, how actually, there is a lot of change between the biopsy and the metastasis, but most importantly, when you compare the tumor to the organoids at the clonal or at the subclonal level, which is the one in blue and pink, you can see how the organoids are absolutely indistinguishable from the parental tumor. And again, we need this to be the case in order for us to be confident in the type of precision medicine solutions that we want to find. So we can perform drug screening in this type of models. We can do this at different concentration. We can do this for single drug, for drug combinations, and we can identify differences in how different patients respond. One of the first things that we looked into is, well, we have this unique resource, right, with this very, very large drug screening. Can we look at how different sarcoma subtypes respond? And so, for example, you can see here how patients with osteosarcoma respond to everolimus, which is an mTOR inhibitor, which is, it's FDA approved and NCCN recommended for osteosarcoma in certain circumstances. And so it's actually clinically used in these patients. And we can find that there is a trend for osteosarcoma to be slightly more responsive than the rest of the other sarcomas we test. This is another example here. We're looking at topotecan, so chemotherapy for leiomyosarcoma, and we could find that leiomyosarcoma patients are the most resistant to topotecan. Now again, this is not a surprising finding. There is a clinical trial done over 20 years ago in the US showing that topotecan does not work for leiomyosarcoma patients. But the importance is that we have the ability to recapitulate these very well-known clinical features, which gives us confidence in the new findings, right? So for example, we could find that dovitinib was actually very effective in leiomyosarcoma compared to other sarcomas. Now, dovitinib is an interesting drug because it's a drug that has actually been through trials. It's been all the way to phase 3 trial. However, the response rate were, about 10% in unstratified population, like you have no way to tell who's going to respond. And these response rates are unfortunately not good enough for a drug to be approved by the FDA. And so this is a molecule that got shelved, was never approved, even though it's been in patient, and it was reasonably safe and well tolerated. And so what we have seen now, we have a much larger cohort. We still see that leiomyosarcoma tends to have a highest response, but in particular now we have the ability to say that, well, uterine leiomyosarcoma, which is one type of leiomyosarcoma arising in the uterus, is actually the most responsive sarcoma within here. And so the idea is that we would have the ability eventually to use this type of technologies to also help salvage some of these molecules that couldn't get over the threshold of approval when you have no way to stratify a population. Now, having said all of that, I'm going to tell you the opposite of it, which is Yes, we can pick up trends across subtypes, and this is just another example of that. This is sorafenib and everolimus. This is a combination therapy, again, used clinically in osteosarcoma. So when you look at responses where, red and white means more responsive, and then yellow means no response at all, you can see that here, this group of most responsive, I think that is probably dying, but this group of most responsive tumors in the box, they're all tile, they're all osteosarcoma. So we can again recapitulate that osteosarcomas are responding to this therapy used clinically. However, what you see toward the end of the line is these tumors highlighted in green, those are also osteosarcoma, clinically indistinguishable from the responders. However, there is no response at all, right? And so this tells you that even though there are obvious trends, there is always exceptions. And so how do we find a way to help everyone find the best therapy? And that is how we get to our concept of precision medicine. Again, a lot of the precision medicine, as it applies to cancer, is done in the genomic space, right? So you take a tumor, you sequence it, you find an alteration in the tumor that's not present in the normal tissue. And for some of these, you do have a drug that you can give the patient with specific, that is only active on tissue which has that alteration. So these are called actionable mutation, right? Now, there are a couple of problems with these, which is in principle an absolutely brilliant idea. The first problem is that the reality is even when you put together all of the different actionable alterations we have now, and this is just a plot of about a dozen of NCI match trial clinical arms, you can see that the prevalence of this individual actionable mutation is low. Even when you put them all together, you can only account for about 4 to 5 patients out of 10 having an actionable defect. The picture is a little bit more complex than that. If you look on this side, you will see actually clinical benefit, right? Patient that responded. Now, this NCI match trial is one of those genomically motivated trials where every patient that's enrolled in one arm has that alteration. So it's a completely stratified population. You would expect that the patient therefore should respond, right? Well, what you can see in gray is that most patients don't respond, even though they have that actionable alteration. So there is obviously big gaps here. The biomarkers we have are not good enough. But what I always like to say is that for the sake of the conversation, even if this was a perfect picture and every patient with a genomic defect is cured, you're still left with over half of patients that you have no way to help today because they have no actionable alteration. And so that is where this concept of functional precision medicine really comes through, right? So we don't need to know really anything about the tumor. We take the tumor, we develop our patient-relevant models, we screen and let the tumor tell us what works best for each and every patient. Now, obviously, there are a couple of very critical issues here. One is that you have to be quick, right? It cannot take you a year to get to this answer. And the other is you are absolutely and critically dependent on having good models, which I hope motivates why we spend so much time truly characterizing and validating this type of models, as I showed you earlier. Let me just give you a quick example. This is an example of a child who came to the clinic with a suspect osteosarcoma, so bone tumor diagnosis. When that happened, the patient gets a biopsy, and we could get to diagnose, and we could get a piece of this biopsy. So this is how the tumor looked like in the child. And again, it's one of these cases where you have tumor cells in a scattered matrix, and this is the models we generated. For this biopsy, we tested about 80 different regimens, and we found a signal of response to platinum, very marked here. The patient went on to receive standard of care, which is a 3-drug regimen. It's a platinum-based regimen. Patients get neoadjuvant therapy for a couple of months, and then they get to surgery. At the time of surgery, this is what was left of the tumor, which is nothing, right? So this was brilliant. At resection, there was 99% of necrosis, which in osteosarcoma pediatric population, the percentage of necrosis that is found after neoadjuvant therapy is actually an almost perfect, prognostic biomarker of response. So patients that have high levels of necrosis end up doing really, really well, decades after surgery. So fantastic response. Now, why do we care if a patient responds or not to neoadjuvant therapy? Well, the reason is that although everyone gets the same regimen, about 30 to 40% of children do not actually respond at all. So they will go receive this 3-drug treatment for 2 months, and then only at the time of surgery we have the ability to see if there was a response or not. So we wanted to look further into this issue, and we actually, for the first study, we got 3 different biopsies. For 2 of them, we could see good responses in the organoids, and that correlated really well with the amount of necrosis that was found at surgery, and also with the status of disease. Both of these children are doing great. There is no evidence of disease now 5 years down the line. We had a 3rd case here where we could see a much reduced response to the therapy in the organoids, and this unfortunately correlated to a very low level of necrosis in the patient, which relapsed quickly, less than 200 days from the first surgery. So we are seeing this very interesting correlation. We also looked at another, and very interesting clinical scenario, which is for metastatic and recurrent osteosarcoma. So say the disease comes back, the patient comes back to the clinic, and in the United States, there are over 10 FDA-approved NCCN recommended regimens. So the oncologist now has 10 options, and they have to pick and choose what to give the patient with absolutely no data whatsoever. What we find in our cohort is that these options that are available do work on a percentage of patients. They just don't work on every patient, right? And so the idea is, can we leverage this type of technology to help a doctor make a data-driven decision when there are safe, approved, but too many options, I guess. And, we have looked at the ability of this type of technologies to match clinical outcome in the case of advanced disease. In that first study, we didn't have a lot of data points, but we had about 5 different data points where we tested a regimen that the patient got immediately after surgery. And then the way in which we looked at responses here for advanced disease is time to next therapy. So the assumption here is that as a tumor progresses, the patient will need to move faster and faster to the next line of therapies because things tend to work worse and worse. And so if you are giving them a therapy and they are able to stay on that regimen and not progress for longer than the therapy before, that is considered a signal of biological activity. And so what we could see is that, when the organoids were actually dying very well in culture, that matched with the patient doing better on the therapy. Vice versa, I think this is something interesting, and we don't spend a lot of time on it, but we're thinking about it, is when we see that an organoid is actually sometimes even growing better on therapy, that matched with patients staying for even shorter duration on that regimen. So again, we have both evidence that, both in the neoadjuvant, newly diagnosed case as well as advanced case, we can see these clinical matches. And so this motivated the start of our clinical trial. This PRIMOS trial opened earlier this year at UCLA. It's an organoid-based functional precision medicine trial in osteosarcoma, where it's a 40-patient trial designed exactly just as the data I showed you now. First, we have an arm for, newly diagnosed osteosarcoma patient, where we are trying to really just answer this simple question. Do we have the ability to tell apart which patients are going to respond to neoadjuvant therapy and which not? Now, the immediate, thing we're trying to do is, can we, spare and allow these patients to move on to the next line of therapy quicker? Also, keeping in mind that tumors are not, they are not stones. If you're giving a therapy that's not working, that does not mean that it's not inducing changes in a tumor. And so we want to try to avoid that. And then in the second arm, we are looking again at this problem of recurrent and metastatic osteosarcoma, where there are 10 or 12 options. Can we pick the one that's better for each patient? And we have the ability to give these results back to the oncologist through our tumor board, and then, of course, match that with long-term outcomes. This is something we are, of course, very passionate about, but the other thing we found in the data set we tested is that When you're screening broadly for drugs which are approved on this type of tumors, you can actually find something that works in over half of the patient cases we screen. The problem is that even though those drugs are FDA approved or NCCN recommended, they are approved for other cancer types. And this is truly the reality for a lot of, the rare cancer space. And so there may be good options out there. It's just, we don't necessarily know that. And the other is, at least in a country like the United States, accessing this type of drugs, off-label, etc., has all sorts of potential additional issues. But we definitely see that. So, in terms of moving forward, we are, of course, from a regulatory standpoint, we have a few different things to do on our end, like getting this pipeline to be CLIA/CAP certified. We have to expand the clinical trial portfolio to really confirm clinical efficacy and the ability to prognosticate. Access to drug, as I mentioned, may be an issue here, but the one thing I did want to leave you with is something we think about a lot. For any type of technology, including sequencing, when you are relying on getting a piece of tissue to analyze, you are highly dependent on which tumor you are sampling and maybe which part of the tumor you're sampling. And you can have a case of a large primary tumor, you're getting a biopsy, what tells you that that piece of tissue is representative of the whole, or you can have cases like our osteosarcoma, or anyway, sarcoma, where you have a lot of metastases in the lung. This is actually a child in our study. And so, you can think that maybe a surgeon goes in and wedges out and takes out this biggest, metastasis because it's creating real clinical problems. And then if that gets to the lab and we test it and we find some agent that works, what tells us that that is going to be the same response for all these other tumors? And we have evidence that there is a lot of heterogeneity. When you're testing 3 samples from the same tumors, you can find, you can find individual sensitivities, and some are shared, some are not. But we truly need to know and understand heterogeneity in the context of rare cancer and sarcoma better in order to be confident that the decisions we want to make are going to be helpful to the patient as a whole. The other thing we're doing in this respect is one of the ways we are trying to think about heterogeneity is we need to be better at understanding it on an experimental level. And so instead of testing 1,000 organoids and getting just one value, one average value, what if we can actually now drill down and do single organoid analysis? Can we then be able to differentiate, which organoid within a patient, population is responsive or not, and what are the features of those that are not responsive? This is one way we do this. This is using a type of microscopy that's called high-speed live cell interferometry. It's actually measuring the weight of the organoids. And so you can see how they're growing and moving around, and then we can detect the rare clone, the rare organoid that is resistant to very effective interventions and analyze that. I don't have really much more time, but this is just another quick way that we're doing that, is by applying a method called cell painting, which is allowing us to stain different structures within organoids. And now, not only say, at an individual organoid level, how organoids are responding, but even at a single cell within organoid level. This is going to allow us to kind of open a new frontier of understanding responses and heterogeneity better. And since I'm out of time, I'm going to stop it here. Just summarizing and telling you that we have this simple, robust, automated platform to do patient-derived tumor organoid work, to grow personalized models with a very short time frame within, from surgery to the results. We spend less than a week. We can do personalized prediction. We can look at landscape analysis. We want to really and truly deliver point-of-care solution. And we're doing a lot of work, including having immunocompetent platform where we can add the immune system. We can test CAR-Ts and see which, for example, product works best for which patient, which is also a big clinical issue. I'll close it here, thanking my lab and the funder, all of the patients participating, and thank you for your attention.
Thank you, Alice. Since you're running off the stage, we'll take questions later during our panel session. So I'd like to warmly welcome another California state representative. So Dr. John Crawford, he is medical director at the Children's Hospital in Orange County and Neurosciences Institute, and he'll tell us how he's been using multi-omics and functional precision medicine for his patients.
Thank you very much for the warm introduction, and thanks to the organizers for a wonderful meeting, and what a beautiful country Latvia is. When I go home, I won't tell my Californians about it, because otherwise you're going to have emigration to Latvia. So we'll keep this a secret, I promise, although it'll be on YouTube, I guess. My only disclosure is I love Star Wars. I'm going to give you 10 years of work in about 15 minutes. So if you blink, you might miss a slide or two, but that's okay. I'll try to get my point across. So I'm a child neurologist and a neuro-oncologist, so I don't know how many neuro-oncologists there are in the room, but I'll give you a brief little overview about brain tumors and why I think this is important for functional precision medicine, since we don't have many standard of care options, particularly for those patients that fail first-line therapy. And then we'll discuss some of our work that started in San Diego over a cup of coffee that has really grown now and has led to clinical trials using functional precision medicine for recurrent medulloblastoma and ependymoma, and I'm hoping that we can do it in diffuse midline glioma very soon. And I'll be hitting Alice up for organoids as well too. So we'll see you at our next tumor board. I'm really happy that functional precision medicine is really making a comeback here. I've seen this in my 20-year career with other things that we think they're dead. I remember hearing that neuroimmunology would never happen. We can't have vaccine therapies. This was 20 years ago. Look at where we are. So I think as we're redefining functional precision medicine in the molecular era, we really have to put it in the context. And I'm very happy that Anthony Latai, who really has been a pioneer in functional precision medicine, was very recently named the director of the National Cancer Institute. And so I think there are wonderful opportunities ahead in this field. And we have to remember it's really important to see the forest through the trees. I never really understood what the statement meant until I came to Latvia. We don't have trees like this in the United States. You really can see a forest through the trees. So I think as we're thinking about approaches here, really it's all about the patient at the end of the day. So how common are brain tumors in Latvia? Well, there are 344,000 children, and at 4 to 5 per 100,000 per year, there are only 18 brain tumors per year here. And this was highlighted in a study from our Latvian colleagues that over 16 years they found 26 cases of medulloblastoma. To put that in perspective, between San Diego and Orange County, we probably have 20 between us every year. So this is a very, very rare disease. There are many different challenges of brain cancer therapy because it's the brain. We have a blood-brain barrier. We have cells that don't exist in other parts. We have toxicities of medicine. And because of that blood-brain barrier, we're not able to achieve our goal in many cases. And we don't even know if the drugs that we give, particularly the biologics, actually hit their target. So primary brain tumors are geographic in location. There are certain types of tumors that live in different parts of the brain. They can present in many, many different ways, and I've spent a good part of my career studying delayed diagnosis of brain tumors and the factors associated with them. They are very rare compared to other cancers. However, brain tumors represent the most common tumors of childhood. And this is something that, because we have collected data through the Central Brain Tumor Registry of the United States, this is really astounding that even given the rarity, it's one of the most common forms of tumor. And most brain tumors in children are malignant. And I remember early on in my training learning that most pediatric brain tumors are non-malignant. That's actually not the case. In older children, you can see that, but in the younger children, the majority of them are malignant, and it's a big problem. Not every child with a brain tumor carries a poor prognosis. It really depends on what the biology of the tumor is. And so we've made great strides in some tumor types. Medulloblastoma, for instance, the Wnt subtype is almost nearly universally curable. And as we think about our treatments, we just don't want our children to survive, we want them to thrive. And so now we are getting into the late effects era where we're worried about the secondary effects of the chemotherapies and the drugs that we give, and this is really important. And this is my then 13-year-old daughter wrote this little picture for a review article, so it makes me smile. I paid her $100 for that, by the way. So the pediatric tumors and adult tumors are just different beasts. The majority of adult tumors are not located in the posterior fossa. About a third of pediatric brain tumors are. And children aren't little adults with regards to brain tumors, the biology, or the responses to drugs. And so when you look at comparatively to the adult tumors, it's just a completely different entity. There are some tumor predisposition syndromes in neuro-oncology, and this was the old classification that I had to study way back in the day. Wasn't this so nice? You could just list everything on one piece of paper. And now with the explosion of genetic information, the 2021 WHO classification is now 2 pages with words you can't even read because they're so small. But that's the point, is that we've gotten more sophisticated with our diagnoses. And really the future of neuro-oncology is not just individual molecular-based therapies, but also, I believe, functional precision medicine. And the great thing about functional precision medicine is that it's not one size fits all. Actually, using whole exome sequencing, targeted panels, not very frequently do you pick up a mutation that's actually actionable where you have a drug. When you're thinking about functional precision medicine, that can apply to every single tumor type, and that could be applicable using multiple different models that could be used in multiple different countries. And it also gives you the benefit of identifying drugs that perhaps could be repurposed that weren't used, and that takes out the whole drug companies having to design a drug when there may be some drugs already that can show some response. And the complexities of brain tumors are enormous. Genetic, epigenetic, transcriptional, post-transcriptional, metabolic, immune regulation, tumor microenvironment. They're complicated. Kids are roses, not onions. But we do have patients that respond to therapy, and we need to know so that we can learn what makes a patient a super responder. This is a patient that I treated that had a low-grade V600E mutant glioma. This child was in a wheelchair, failed standard of therapies, and after about a month on therapy, I couldn't catch the child anymore with a near complete response in MRI. Never in my career did I think swallowing a pill could shrink a tumor, so we are in a very exciting time. There are times when chemotherapy tumors respond to chemotherapy. This is a patient with a low-grade brainstem glioma where carboplatin vincristine just melted the tumor. That doesn't happen all the time. This is a patient of mine with diffuse leptomeningeal glial neuronal tumor, and you can see on the left-hand panel all the white sugar coating here. And then after about a month of a new pan-RAF inhibitor, tovorafenib, this disease completely melted. I've never seen anything like this in my career. This is a 7-year-old boy with what would have been termed a glioblastoma back in the day, who probably would have had multiple chemotherapy regimens and radiation therapy. I've seen these patients in my clinic in wheelchairs with G-tubes and trach tubes. This patient, all he had was a surgery and an oral biologic with a TRK inhibitor, and 6 years out of therapy, he's only on that drug, never has seen radiation, never chemotherapy, and he's perfect. So now I'm going to get into why I was invited here, which was novel approaches to brain tumor therapy. As I said, about 15 years ago or so, Rob Weschler and I had a discussion over coffee about what can we do differently to treat brain tumors, and then we brought our neurosurgeon in, Mike Levy, because it's critical to get tissue And you'd be surprised, back in the day, the tissue would just go right down the drain. So I'm so happy that we're actually collecting every little ounce of tissue, blood, spinal fluid, et cetera, to help our patients. And it really started out with xenografting and with using drug panels from the Sanford Burnham, about 3,000 drugs in a library. And then we thought, I think it's time to bring it back to the patient and not use xenografts. And so we collaborated with the University of Washington, where we were doing drug screening panels, and that is what's led to clinical trials, which we're very excited about. And this concept of this molecular tumor board, it really started with Inese, and it's amazing how this caught on like wildfire, and everybody wanted to be part of this group, whether they did primary research in neuro-oncology or not. It's not a hard sell to bring basic scientists in with a deadly brain cancer. And so it's just an incredibly talented group. This is our workflow here that we're putting together for publication, where the standard is whole exome sequencing of the tumor, RNA sequencing, whole genome sequencing from the blood, methylation analysis, gene set enrichment analysis called Discover analysis from RNA sequencing, and then our drug screening as well too, that I'll go over in a minute. This is the characteristics of our patients. The majority of them were recurrent tumors, and the majority of them were high-grade gliomas, mostly diffuse midline gliomas. And what I'm most proud of is that we accepted all comers. When you look at the insurance information, this is a really diverse group, and the insurance matches what the insurance status is at our hospital. So it really was an inclusive study that included all patients. And I'm very, very proud of that. And we have to be responsible as we think about clinical trials and accessibility to therapies. And this largely was a feasibility trial, and this shows you the number of days in order to get the results back. And what you'll see here is actually the high throughput drug screen. There's a lot of work involved with that, but that was the fastest one to come back. We beat methylation and whole genome and whole exome sequencing. It was really impressive how fast we could get those results back, and that's really, in part, what had led to clinical trials for recurrent medulloblastoma and ependymoma. So we had a cohort of 45 patients who underwent a variety of testing. 6 of them had a change in diagnosis, 34 of them had a refinement in diagnosis. And then when we get to the management of patients, 19 patients had a change in management. Whether that was due to 1 result, 2 results, or 3 results, whether it was the molecular testing or the drug screening or a combination that led to changes in clinical management, I think this is pretty profound. And to my knowledge, this is the largest series of a functional precision feasibility trial in pediatric neuro-oncology. We were so blessed to have Ken Aldapi at the NIH perform all of our methylation analysis. And methylation is really critical to get you, what I say, the right diagnosis. I think that that's a really impressive technology that is really leading the way in neuro-oncology, but it's not functional by nature. It'll get you the name of the diagnosis. Here's a list of our somatic gene sequencing of our patients characterized by tumor type. And what was surprising is germline variants were identified in 20% of the cases. Again, early on in my training, there was this notion that brain tumors are not congenital and the germline isn't important. And increasingly, we're identifying that in fact is the case. And that is being studied in a much larger cohort at the Children's Oncology Group with the APEC clinical trial. Another novel-based therapy that we used here is Discover analysis, and we were very happy to have Jill Meseroff and her team help us out here. And really, this is looking at 150 overexpressed genes using a gene set enrichment analysis to be able to predict, based on RNA sequencing, responsiveness to drugs. And this is just an example of a patient of ours where Discover analysis had led to the proposition of 5 drugs. What I'm most proud of is the design of our custom gene panel. So Rob Weschler-Ray, in conjunction with a lot of us neuro-oncologists in the country, came together and put an initial 175 custom gene panel together. This number has grown over to 200 now, and this includes drugs of a variety of different mechanisms of action. Some are FDA approved, some are investigational, and there's 2 pages here. And basically, what you get is you get concentration curves. So we have 5 different concentrations per cell. They're done in duplicate, 2,000 cells per well, 3-day incubation with cell titer glow assay results. And what's very impressive is you can do the drug screening on minute amounts of tissue that could be obtained from a stereotactic robotic guided biopsy. And this is really important when we're talking about diffuse midline gliomas of the pons, an incurable tumor. And I look forward to working for the next clinical trial that will use functional precision medicine as an upfront therapy for diffuse midline gliomas. This is an example of our drug screening platform. We have an online portal where you can query by drug type, mechanism of action. You can get these heat maps here. We're looking into advanced drug screening predictability scoring with a blood-brain barrier score. As I had mentioned before, these drugs aren't crossing. And there's also the drug screen sensitivity scoring. And hopefully AI can help us with predictive algorithms in terms of what drugs will most likely enter the CNS. Oh boy, I didn't do it, I swear. This is an example of recurrent posterior fossa ependymoma, some drug screening results that we actually used in a patient. Sorry, let me go back. There we go. And this is an example of a patient with diffuse midline glioma. This was published a couple of months ago, a concentration curve, and I selectively picked out 6 here. And in addition to ONC201 therapy that the patient received, disulfiram, which is used to treat alcohol abuse, was a very impressive response. And after discussion with the family, we decided to do combination therapy. He had a very nice response to therapy with regards to a combination of ONC201 and disulfiram, but the toxicity was pretty profound. It caused a sensory neuropathy, and this is just case in point that we have to be careful when we're combining drugs, particularly ones that haven't been studied before. But what I've always found in the neuro-oncology community is kids and parents really want to do what's best, and they're willing to take the risks. I started my training when we didn't biopsy the brainstem ever, and now in many places, including ours, it's standard of care so that we can really push the field forward. So, in conclusion, functional precision medicine is feasible in both the upfront and recurrent setting. We really need predictive algorithms to assess blood-brain barrier penetrance and the prediction of efficacy and toxicity. As I mentioned, studies are underway in recurrent medulloblastoma and ependymoma, and I hope diffuse midline glioma is next. And future studies will utilize combination drug screenings as opposed to single drug, AI approaches, and after Alice's talk, we really should reconsider organoids as well too. This is an enormous team. I'm so proud to be part of this team as the lead clinician, the one who gives the drugs to the patients, but really all of the basic science and great minds are behind the scenes, and I'm incredibly grateful to have this team. This is my family. Sorry, the first family slide I've seen, but I wouldn't be here without them, so I thank them. And my last slide, ever since 2009, I have kept track of every single patient of mine that's died of a brain tumor. And each one of them is represented here as stars in the sky. I think we can all agree that is way too many stars in the sky, way too many children lost. We have a lot of work to do, but really the field is extraordinarily bright. It's international, collaborative, and I look forward to great things happening in pediatric neuro-oncology in the years to come. Thank you for your attention.
Thank you very much for the very inspirational talk. And yeah, there are too many stars. Now moving to the Europe and talking about the clinical trial, I'm happy to introduce you with Thea Pemovska. She's a senior postdoc researcher at Department of Medicine at University of Vienna, Austria. And she'll be talking about the functional precision hematology, the Austrian experience.
Yeah. How does it? Oh, okay. Sorry. Yeah, thank you, Inese, for the kind introduction. I also want to thank you for inviting me to this great meeting, and I'm really excited today to sort of end off this session and really reiterate some of the messages that were already said, but also introduce how we've approached functional precision medicine in Austria, primarily for blood cancer patients. So, the promise of precision medicine has was basically to identify historically static features, so genomic mutations or expression patterns as unique characteristics of the individual patient's tumor, and then use this information to identify therapeutically targetable drivers. So the core strategy is matching this molecular information primarily to corresponding targeted therapies based on molecular profiling and biomarker information. However, the reality check is that at least this aspect of precision medicine has not really lived up to its promise. As we've heard earlier today from several other speakers, these genomically driven matchings to therapies are only possible in less than 10% of cancer patients. This has been shown in multiple large trials like the NCI match that Alice talked about earlier, but also the Shiva trial, Moscato trial, Winter trial. There are several others that for less than 10% of patients, we have really clinically validated approved therapies that can be matched to their mutations. But even worse, for 5% of those patients, we can actually see a clinical benefit. So a really small proportion of patients derive a meaningful clinical benefit solely based on genotype-matched therapies. And yet, we are still faced with 20 million new cancer cases globally and 10 million deaths annually, largely due to ineffective therapies. So we clearly require additional information to match therapies to patients. So what are some of the reasons? Some of these things were mentioned before, but just to set the stage again. So why genetic information alone often falls short? So in our endeavors of sequencing practices, we frequently come across not yet druggable mutations. So these are primarily mutations in tumor suppressor genes that are loss of function, and drugs are blocking a function. So we actually need activators of these drugs. And a lot of these mutations, like TP53 and so, are, of course, so oncogenically important, but still remain therapeutically intractable. Even when we can match therapies to patients based on their mutational profiles, the response duration may be limited or not durable due to a plethora of resistant mechanisms. And last but not least, we still perhaps underappreciate the biological complexity that exists, mainly due to cellular plasticity, tumor heterogeneity, and different compensatory signals that confound these simple genotype to phenotype predictions. So it's clear that we require additional information to make the best treatment decisions. So Unfortunately, this initial promise that started over 20 years ago with some early successes like imatinib that was introduced by the keynote speaker earlier, precision oncology has not lived up to this initial promise. It is mostly practiced today. So this classical model of matching individual mutated driver oncogenes with their specific inhibitors is not that simple in practice. We also have many targeted therapies that demonstrate efficacy despite not having a clear genetic biomarker guidance, highlighting gaps in our understanding. So enter functional precision medicine. So this was introduced by the prior speakers in this session. So the core philosophy is really put drugs on the cancer cells and see if they work. So it's a direct approach that directly measures how the cancer cells of individual patients respond to a plethora of drugs, and by that provide immediate actionable therapeutic insights. So how it exactly works, so this is providing a direct perturbation where patient tumor cells are directly exposed to therapeutic agents in a controlled laboratory conditions ex vivo. There can be different ways of doing this depending on tumor types. I will explain a bit later how we approach it for blood cancers. This leads to dynamic data generation where we get really functional real-time data encompassing key cellular vulnerabilities and dependencies. But most importantly, functional precision medicine does not really care about what the mechanism is in the first instance, right? So we want to read out how do the cells of an individual patient respond to drugs. What these cells express or which mutations they carry, it's not so important in the first instance. Of course, later to try to understand why certain cells responded to these drugs to others, having additional molecular data is helpful. But at the very first instance, the approach can be very agnostic with respect to disease mechanisms and can focus on observable cellular responses. Last but not least, functional precision medicine can lead to immediate translation of results, that we've seen some examples given by the prior speakers, and this has potential to provide personalized treatment recommendations directly back to clinicians and patients in a rapid turnaround time that is clinically relevant. So we often know that cancer patients cannot wait forever for the next treatment strategy, and tumors are particularly aggressive in certain instances. That's definitely the case in certain blood cancers. And it can provide evidence-based precision where functional evidence for therapeutic selection beyond genetic prediction alone. But last but not least, of course, I'm not excluding genomic profiling, but functional precision medicine can be a way of genomic interpretation and tell us which genetic alterations truly matter therapeutically. So how we wanted to approach this questions of how can we introduce functional precision medicine in clinical routine for blood cancers. So we first had developed an imaging-based functional precision medicine approach at the Research Center for Molecular Medicine in Vienna, Austria, in the research group of Giulio Superti-Furga, and then collaborated with the Division of Hematology at the Medical University of Vienna, where we took, I'm really sorry for the quality of the slides, for me on the screen much better, but so we isolated peripheral blood, bone marrow samples or lymph nodes because the trial was open to both leukemia and lymphoma patients. Process them to get single-cell suspension, so for bone marrow and peripheral blood, doing a FICO gradient to isolate the mononuclear cells, or then tissue sort of dissection for the lymph nodes to get single-cell suspension. We seeded the cells on top of already pre-drugged plates that were prepared by acoustic dispensing, incubated them very acutely, similarly to ALICE, so up to 24 hours, then stained with marker-specific fluorescent antibodies antibodies that were communicated to us from the laboratory medicine or pathology departments that were specific to the biopsy, and then performed high-content single-cell imaging. Now, since we had in the same biopsy both cancer cells and microenvironmental cells, this allows us to score for differential cytotoxicity, really prioritizing agents that have a cancer-selective activity. These results were then discussed in a multidisciplinary tumor board, and treatment recommendations were communicated back to clinicians and patients. The endpoint of this trial was progression-free survival ratio. What does this mean? We looked at the progression-free survival on functional precision medicine guided therapy compared to the progression-free survival on their most prior treatment, because the expectation in cancer, especially in the relapse and refractory setting, is that with every subsequent therapy, the duration of response gets shorter and shorter. So if you're able to extend this time frame, this is considered a benefit for the patient. And this endpoint is becoming more or less standard for a lot of precision medicine trials, both genomic and functional. So in this, I'm really sorry for the slides on my screen, it's much better, so I don't know what happens. But we included roughly 200 patients that were screened. Of those, 143 underwent this functional precision medicine testing, so 74% of the patient population. 76 patients were evaluable for outcome measures, of which 56 received SCF-guided therapy, or 39% of the patient population. 20 patients were kind of handled in the same way, but received physician's choice therapies and were used for a comparison of outcome in a post-hoc analysis. So importantly, 30 patients achieved an improved PFS ratio above or equal to 1.3, with 11 patients marked with stars having an ongoing response at the censoring date, with a median follow-up of almost 2 years. And then most exceptionally, we also identified 12 patients, or 40% of the responders, that had what we call an exceptional response. So really almost 3 times more than the expected duration of response for their respective disease entity. And I'm not sure you see it, but the patients that mostly benefited in this first trial were lymphoma patients, and particularly T-cell lymphoma patients. In addition, patients that were more fit benefited more from functional precision medicine guided therapies, so patients with ECOG 0 or 1. This was, in the end, the first sort of prospective trial that evaluated a functional precision medicine test to deliver N01 therapies in Vienna. So the results of this trial really encouraged us to set up a follow-up trial called the EXALT-2 trial. This is then now a randomized trial where, in a head-to-head comparison, patients get randomized in functional precision medicine arm or comprehensive genomic profiling arm, which was delivered by Foundation One Heme from Roche Diagnostics, and then physician's choice. In a 4-to-4-to-2 randomization patterns, because it was allowed for arm switching when a report was technically not feasible to be delivered in either the functional precision medicine or the genomic arms. And initially we had a lot of switches to physician's choice, so we revamped the randomization to balance that. Importantly, both assays are performed for all patients, but the results are blinded, so only the results of the arm in which the patient is randomized in are then looked at in the multidisciplinary tumor board, which is supported by the Navify tumor board software, also from Roche Diagnostics, and that was primarily the interest of Roche for being involved in the trial. Our tumor boards are attended with at least 2 hemato-oncologists, so that there is a consensus in treatment decision-making, a pharmacist that looks at the drug interactions, especially when drug combination therapies are recommended, a pathologist or expert in laboratory medicine that can confirm the diagnosis, that can confirm that there were cancer cells really present in the biopsy, and also can confirm that the staining pattern that we used is actually corresponding with the expression profile of the sample in the clinic, and a molecular biologist like myself to give further insights of the test results. Then a treatment recommendation was given and patients were followed up until progression. This trial is open in 5 medical university hospitals across Austria. It started in Q2 of 2020, so right in the heat of COVID. So in the beginning it was very tricky with recruitment, but we slowly picked up. The trial is sponsored by the Medical University of Vienna, financed by Roche, and the PI is Philipp Staber and Lukas Kacianka. So the inclusion criteria are, the trial is open for relapsed and refractory aggressive hematological malignancy with at least 2 prior therapy lines or 1 therapy line with no further standard of care is available. The response to prior therapy should be less than 1 year. The clinical performance status should be for fit patients. Anti-tumor therapy should be medically feasible, and real-time biopsy should be doable. Exclusions: secondary malignancy, participation in another clinical trial, pregnancy, unfit patients, Hodgkin's lymphoma, and pediatric patients. The efficacy endpoints are individual PFS benefit or PFS ratio on each of the arm compared to the PFS on the most prior treatment, average PFS ratio on each of the arms, overall response rate, and number of treatable targets. Secondary endpoints, overall survival, successful bridging to stem cell transplantation for eligible patients. As we know, for many blood cancers, this still remains the only curative option. And time to result from informed consent, biopsy, availability, randomization. So at the same time also feasibility. So we started also with the imaging approach in the beginning, but in the meantime, the people doing that did a startup, and then a lot of the samples were rejected. So we ended up setting up our own platform based on high-throughput flow cytometry in the lab, and we are testing roughly 140 different drugs approved in Europe, and using the IQ-free automated high-throughput cytometer, and we quantify a marker surface expression, cancer selective killing, and clonal drug response tracking. The current turnaround time is roughly 3 to 4 days. Oops. This is a patient journey example that I wanted to share. So this was a 37-year-old male that was diagnosed with T-cell rich B-cell lymphoma that failed 4 prior therapy lines, and in January 2022 was enrolled in our EXALT trial randomized in the drug screening arm. The results showed that the patient responded very selectively to a PRP kinase inhibitor, copanlisib on the left, and vandetanib, a VEGFR inhibitors, both not at all approved for this disease entity. But the tumor board decided to recommend a combination of tafasitamab, an anti-CD19 monoclonal antibody, and either copanlisib or vandetanib. The patient went on tafasitamab and vandetanib, and 2 months later, the evaluation had already a partial response and evaluation for stem cell transplantation. So this is the PET-CT scan before receiving any therapy, restaging, which was a partial response, so really significantly regressive uptake in almost all previously known affected lymph nodes, and then after 4 months, really a regression in size in most lymphoma manifestations, so a complete response, and the patient then was able to undergo a stem cell transplantation and is still disease-free several years later. And just for comparison, we also have performed a Foundation 1 heme assay for this patient, which didn't identify any mutational events. So had we just relied on the mutational events, this patient would have not been able to get treated with anything, and it's amazing how it went. Earlier this year, we published the feasibility analysis from the EXALT-2 trial, and an interim analysis was not planned per protocol, so no outcome data, but only a feasibility analysis of 56 patients. So we screened, yeah, you cannot see anything, I'm sorry. We screened 56 patients, of which 54 could undergo a real-time biopsy, so 96% of these. 77% or 43 patients could be randomized, and this was our intent to treat population. 42 patients could receive EXALT-2 tumor board recommendations, so 98% of the intent to treat population, and 29 patients or 68% of the intent to treat population could undergo treatment either guided by functional, genomic, or physician's choice. Similarly, like John showed, our time to report was the quickest with the functional precision medicine approach, with a median time of 6.5 days for the flow cytometry-based, 7 days for the image-based, and close to 19 days for the genomic precision medicine arm. Most importantly, we technically, possibly a result was available in 64% for the imaging, 86% from flow cytometry, and 86% of the genomic. Obviously, a drug screening assay reveals a target in 100% of the cases, where with the genomics it was 76%, and therapy was feasible in 86% with the flow cytometry, and 64% for imaging, and 65% for the genomics process. When we looked at the results of patients that had both genomic and functional precision medicine tests, the top hits or targets overlapped in 60% of cases. So in conclusion, I want to show, hope I showed you, and also the other speakers in the session, that functional precision medicine can be, it's feasible to integrate it in clinical routine as a clinical decision-making tool for advanced aggressive blood cancers, but also sarcomas and also pediatric brain tumors. While we all have published or have shown anecdotal successes, we really require high-level efficacy evidence to really make functional precision medicine routine in clinical practice. And last but not least, we have a plethora of medicines that exist out there, and this approach really allows us to discover unexpected mechanistic and clinically relevant opportunities, and really highlights the value of searching for novel indications for all drugs. I want to thank the patients and their families for participating in our studies, the Staber group for their work, as well as the Department of Hematology and Pathology at the Medical University, and our partner hospitals and our fundings. Thank you very much for your attention, and sorry for the slides. I should have checked them earlier.
Thank you very much, and I apologize for the technical problems. It looked very good here. Yeah. So I invite all our speakers from this session to the stage for panel discussion. And if there are questions from the audience, this is the time when we could address our speakers. If somebody has the questions, please let us know. I could start with the first question. John. Yeah, I know. You are fast. Oh. Okay.
Oh, I can sit on the floor.
No, I can bring it up. Okay. So, John, you kind of mentioned that, but I didn't get it like this really. How do you get enough cells from the brain tumors? Because, like, if you have to test like 120 drugs.
Yeah, I mean, it's 2,000 cells per well, so
Duplicates.
And duplicates, yeah. We've just been lucky with enough cells, even with the needle biopsies too. The number of cells went from 2,000 probably down to 1,000 per well, but we've been lucky. We've never had a case where that's not true. When we first started it, especially with the brainstem biopsies, we only sent 2 cores from the brainstem, and that was the only time that we didn't have sufficient cells to do the drug screening. So now routinely we do 4 to 5 cores of tissue, and it's actually relatively safe, you know, knock on wood. So yeah, we've been lucky.
Thank you.
So one thing I noticed about all 3 of the functional precision medicine platforms that were presented here is you've been really great at quick turnaround time, so a week or less. But perhaps not all drugs work that way, and I'm wondering if you can maybe discuss ways to try to account for drugs that maybe work rather than through cell-killing effects, and are you thinking about ways to address that?
Yeah, maybe I can start. Definitely, and I mean, there is obviously flexibility in that we can adjust the timeline for drugs that work differently, but also what we're putting a lot of effort in is, you know, a cell may not be dying immediately, but they are nudged, there is an effect, and can we find a method that allows us to measure that effect early on? Because, you know, the timeline needs to stay quick, to stay quite, you know, quite rapid for this to be clinically meaningful. So that's what we're looking at. The other thing is, because once you start to develop these very, very large data sets, right, you also have a lot of, I think the biggest value is not measuring a single sample. The biggest value is the data on all the other samples we have. And so we actually normalize, we have ways to normalize the effect for every patient, for the average of every other patient tested against the same drug. And so again, that allows you to really normalize for drugs that are overly toxic, specifically, as well as for drugs that don't necessarily cause, you know, overt cell death. But again, it allows you to pick up those, you know, highest responders within the range. And there the power is really, the more data you have, the better you're positioned to make, you know, earlier conclusions, yeah.
Yeah, if I can add, I think for us it was more, we wanted to really prevent that any divergence occurs in culture from the original sample or this. And also I think a lot of it depends also on the readout. So I think both speakers have used cell-titer glow, so maybe it's really on this proliferation heavy also mechanism of action of drugs that you can score well. But I think with some of these imaging or even flow cytometry things, for example, we've seen responses to azacitidine or things that are typically expected that they take longer to give activity. So I think also things should be looked at. It's a fine balance in the end of what is prioritized more, but I think in the end of the day, as Alice said, we have to validate our platforms as best as we can and juggle sort of authenticity of the sample or the integrity of the sample and the turnaround time so that it's still clinically implementable. I mean, in the blood cancer space, if you remember, Caroline, we've seen within 7 days, a patient had a completely different drug screening profile and completely different molecular profile. So in some diseases, you just cannot afford to screen very long or to do if we're aiming to really make this a routine practice. So yeah, I think it's a balancing game.
There is a question, yeah.
Thank you for all the great talks. One thing which you a bit kind of were touching already, but what I think that is very important is the feasibility of the studies and what is actually something, when you have, for example, functional precision medicine study ongoing, what do you actually prioritize and who is doing the prioritization if you don't have enough of sample, like whether it's RNA sequencing, whether it's exome sequencing, and then something which then comes also to the pipeline. So drug testing itself is very fast, but do you actually really do every single time the characterization of these PDCs or patient-derived cancer cells or organoids, whatever you use, to be sure that they correspond to the tissue? I think that this is extremely important with the solid tumors. Or do you just trust that you did it, like, you know, for 100 of cases or 50 cases and it worked out, and then you, or do you always validate, and if, how?
I mean, I think the right answer is you should always characterize, to be completely honest. Our gold standard is all genome sequencing, and we can do that, down to 20,000 cells. The approach now with some of the single cell sequencing approaches, you can go even lower, but I think that now, so it doesn't take a ton of cells, we use 5,000 cells per well for drug screening, so again, you're not wasting. I think that's important and that should be done. We also do RNA-seq a lot. I think, there is a lot more variability. It's a little bit easier to make sure that your sample is absolutely validated, if you do deep whole genome sequencing. So that's why it's my preferred method to do that. And then, I don't know.
Yeah, so for us, I've set up our drug plate such that sort of the most approved drugs are on one plate. So in case there is a limited material, we are able to screen, let's say, one plate instead of two plates. Additionally, for the genomic analysis, often when it's particularly a lymphoma case or a lymph node sample, this goes to pathology anyway, and they make slides, and slides are sent to Foundation One Heme for performing the thing, so it doesn't take away too much from the material we would anyway get. So in this way, but of course, being in an academic study, it's always like, do anything and everything anytime. But often I have to sometimes do a hard no, even when my clinician collaborator is really insisting, because I know, we are really screening on this single cell level on a marker cell population, and if the tumor cell content is low to begin with, and then you're additionally hit with a very low cell number, you know that the cell numbers that you'll be scoring will likely not be predictive. So at some point, there is the limit of detection that you should obviously.
Yeah. And I did forget the first part of the question. Yeah. So we prioritize by, we want to do FDA approved for the indication. So depending on the sarcoma type, it's going to look different. And then we talk to our oncologists. We do clinical trials, so things in clinical trial for the indication that are exciting. We look at, new literature, so things that people have been thinking about in terms of mechanism for the specific sarcoma types. And then we talk to our oncologists, so we're always interested in seeing what they feel like they should be seeing. So we add that, and then we move down, if we have enough cells, then you move out of the specific sarcoma type, you go for other sarcoma types, and then other cancer indications. So we kind of do modular. When you do things in a completely customized fashion, it's complicated. It's a lot of extra work. I think it's very warranted. If you ask my people, they may disagree with me, but I think it's important to, particularly in this rare cancer, you need to have the ability to test what makes the most sense, yeah. And now we're looking into, of course, leveraging AI, right, to help us pick the best, and edge our bets when you have a limited number of cells, so.
So I think another big challenge we have now is convincing payers, either European governments or U.S. insurance companies, that these are assays that should be reimbursable. And I'd like to hear some thoughts on how you think that might be possible.
Yeah, that's a great question. There's number one, approving the assay, number two, approving the drugs that the assay demonstrates. So it's really two parts. The University of Washington is a CLIA certified lab. So it's a regulated lab, so they could, bill insurers, but it's been a reimbursement issue. And because of what we've done in our molecular tumor board, there have been families that have been paying out of pocket for this analysis. And that makes me sad because it's not a perfect analysis. You have to take it into the context of the clinical picture. You can't just go with the number one drug that shows up. I don't think it really works like that. It's more complicated. In terms of the drugs, our panel now includes more investigational drugs as well, but thus far, I haven't had any problem getting the drugs approved as long as they're FDA approved, and almost all of them are. It's just a time sink. It's a lot of denials, a lot of peer-to-peer, but I have to give credit in most of the cases. People have approved these drugs, and we did a PMNET trial on DIPG that was molecular guided, not functional, and it was the same issue, a combination of 3 to 5 drugs. You got to get through insurance, and we didn't really have any problem getting it through, so we were lucky, but I know that's not the case everywhere across every country.
Yeah, for us, I didn't have time in the lecture to say, so the way we are also managed to run our trial is, so when a recommendation happens, and this drug happens to, of course, not be often indicated for the particular disease or diagnosis that the patient has. So first, there has to be with basically an official document from the tumor board that this is the recommended regimen. This goes to the medical directorate of the hospital that has to approve it. And then often, if this is, let's say, anyhow prescribable within the hematological blood cancer space, then okay. If not, then typically the route has been that we approach the pharmaceutical company that develops this drug to provide this drug for a 2-month basis, if as a gift, basically. Then if the patient experiences benefit on this treatment after the 2-month period, the health insurance picks up the tab further. And this is not, unfortunately, it's not a procedure that is pan-European. So that's why it prevented us to open up sites more internationally. And then to the second aspect of like, what would it take in terms of reimbursement? I think there's a third model, as I'm finding out at some other training. So one is also to look at what existing codes exist. So I don't know under which codes the genetic tests may be approved. So if there is like a code for aiding treatment decisions, then maybe you are able to piggyback on that despite of technology. Or like in our case, if there's anyway flow cytometry type analysis, maybe you're able to piggyback. But this again would be country specific and not region or even continent specific. So I think there's a lot of work still to be done on the regulatory side to unify rules to make it quicker and more transparent what the procedure would be and what type of evidence would be required. Yeah, so I think at least on the European side, there have been talks that we want to do a more multi-country trials and also maybe performing assays in different laboratories to really verify that it doesn't just work in your lab, but across things. And I think also automatization would definitely aid in this process. But yeah, it's a hurdle. And also the last thing is, thank you for you being here. And unfortunately, many clinicians outside of the heme space are not so open, maybe in the pediatric, because of course it's a very critical population. But a lot of clinicians in oncology, even in our institution, are otherwise not so open to let's say, experimental regimens. And even in the blood cancer space, what I see often, as you say, it's not always the top hit. I think clinicians would still like that a particular regimen fits on a published scheme, whether it's in that particular disease entity or something, because they like to have some support a bit other than the drug test to have confidence to really treat this patient, to know that there won't be, many funny things happening by combining the drugs or whatever. So.
And I mean, I think in the US, particularly for reimbursement, I think the experience is the same for drugs. Like you can access drug as long as it's approved. It's a huge time sink, and I don't think it should be. There should be processes, but we don't have that. However, if you have the time and to invest, you can get it. But the reimbursement, particularly for the assays in the US, is plagued by old language that is 20 to 30 years old. Around, there are basically no coverage determinations that have been written by the CMS and this type of, providers that insurance look at to make coverage decisions, where they say that these type of assays are not good, which was not untrue 20, 30 years ago. And so now a lot of the work that we're doing with the Society for Functional Precision Medicine is really trying to get this outdated language around 20, 30-year-old technologies taken away from this non-coverage determination so that there can be a new one. And for us, once the Center for Medicare and Medicaid says they're willing to reimburse something, that's when then the private insurance come in. And so, it's a layer. You got to go through the process. But yeah, the problem is that we have some old determinations there that are impacting us today.
Mm-hmm. Thank you. I think we could sit here hours and talk about these things, but unfortunately, our time is limited, and I am very, very grateful for really great talks. I really enjoyed every single talk, and I want to remind to you and to the people who are here, there are wine and snacks in the cafeteria right back in the library where we had the lunch, so everybody's welcome for this networking continuation with the wine and snacks, and thank you for being here and being patient and listening our discussions. I hope it will gonna help. Thank you.